Hello, my name is Ygor. I am a master's student and a member of the Brazilian research group Antarctic, Medicine, Physiology, and Anthropology in Antarctica. I will present the work entitled Thermal Insulation Clothes Promotes Increase in Warhead Sweat Efficiency During an Antarctic Expedition. And I would like to thank all the institutions that collaborate with our group. Antarctica is the continent with the lowest temperatures, the windiest, and the driest on Earth. Due to these characteristics, Antarctica is also known for the acronym ice, isolated, cold, confined, and extreme. This way, behavioral responses such as the use of appropriate clothing are essential for human survival. However, the thermal isolation characteristic of the clothing worn by Brazilian researchers associated with long walks and the transport of research equipment create a microclimate between the skin and the windbreaker, with temperatures reaching 29 degrees Celsius, what is considered hot microclimates. This microclimate is enough to increase the production of sweating as well as to increase the internal temperature by 1 degree Celsius during a walk. Due to restricted exchange of heat between the body and the environment. This way, repeated stimuli can lead to acclimatization to heat. Thus, the aim of the study was to evaluate within an Antarctic expedition cold resulting thermoregulatory adaptations to heat. Material and methods. This experimental study followed the rule established by the Brazilian National Health Council and was approved by the Research Ethics Committee of the Universidade Federal de Minas Gerais. Characteristics of the volunteers. 7 volunteers participated in the research, 5 women and 2 men. The age average was 30 years old. The height average was only 171 centimeters. Experimental design. On the first day of data collection was performed an aerobic fitness test, protocol adapted from JITRIT. 60% of the maximum speed achieved in the VO2 max test was used to calculate the running speed used in the acclimatization protocol. On the second day, sub-maximum exercise in a hot environment was performed. This data collection referring to the pre-Antarctic situation. After data collection, the volunteers traveled to the Antarctic continent, where they remained camped for 32 days. After this period, they returned to Brazil and repeated the same procedures as the pre-Antarctic situation. Data collection highlighted to the post-Antarctic situation. Experimental protocol. The submaximal exercise was used to observe the adaptive response to heat. The exercise consisted of a 45-minute run, where the core temperature was measured every 5 minutes. At the end of the exercise, the filter paper of the sweating analysis was removed, and the number of active sweat glands was printed. The incline of the treadmill was 1%, the running speed used was 60% of the maximum speed of the oxygen consumption test. The ambient temperature was 31 degrees Celsius and 6% relative humidity. Measured variables and calculated parameters. Figure 3a, referring to gastrointestinal temperature telethermometer and B gastrointestinal temperature capsule. The local sweat production, TLS, and the number of active sweat glands, ASG, were collected in the regions forehead, arm, chest, forearm tag. The rate of local sweating was calculated using the following equation. The weight of the filter paper at the end of the exercise minus the weight of the filter paper at the beginning of the exercise divided by the exercise time and by filter paper area. To calculate the number of active sweat glands, we use a paper cutout. The paper cutouts were kept in a tightly closed glass container and protected from light, along with iodine crystal for about 48 hours. After exercise, the iodized paper was pressed against the skin for 3 seconds. Lastly, the number of ASG was determined by counting the blue dots printed on the paper in 3 1 square centimeters quadrants previously drawn. Statistical analysis. Equivariance was tested and confirmed in sigma plot by checking the variability between the groupings. A two-way repeated measures analysis of variance was used to compare core temperature measured during the submaximal exercise performed in a hot environment. Student t-test was used for comparisons between averages of TLS and ASG, that are shown as mean standard deviation. Significance level used was P more or less 0.05. Results. The table number 1 represents physical characteristics of the volunteers at pre- and post-Antarctic moments. The column number 1 refers to the pre- and post-Antarctic moments. Number 2, body mass. Number 3, body fat. Number 4, VO2 max. We did not observe statistical difference between the post and the pre-Antarctic moments. The graph number 1 represents the core temperature. The white ball represents the pre-Antarctic situation, and the black ball represents post-Antarctic situation. The y-axis refers to the core temperature in degrees Celsius, and the x-axis refers to the exercise duration 45 minutes. As expected, the core temperature was increased over time. However, we see no difference between situations. The table number 2 refers total local sweat during the 45-minute run in a hot environment. We observed that in the post-Antarctic situation, there was an increase in the production of forehead sweating, but it was not possible to observe difference in the other variables, only a tendency to increase the sweating of the forearm in the post situation. The table number 3 refers number of active sweat glands during the 45 minutes run in a hot environment. We observed that in the post-Antarctic situation, there was an increase in the number of active sweat glands on the forehead, but it was not possible to observe difference in the other variables, only a tendency to increase the sweating of the forearm in the post-Antarctic situation. Conclusion. The use of thermal insulation clothing during 32 days of a camp in Antarctica resulted in an increased sweat efficiency on the forehead of young adults, an adaptation induced by the hot microclimate. We also thank the military personnel involved in the Brazilian Perantar for logistical support. We also thank Professor Andrés Aranque for allowing data collection with the individuals of the research group lead. Very especially, they also thank the volunteers who participated in this study. We also thank the founding institutions because without them this study would not be possible. These are our promotion pages. These are the study authors. Thank you for watching the presentation. This is my email for contact.